When we talk about cell division, we're really talking about two processes put together. So the first part of cell division is mitosis. In mitosis, technically, all you're doing is you're taking the genetic material in the original parent that was copied and splitting it into two locations to create two new daughter nuclei. Having just mitosis take place, though, only creates a single cell with two daughter nuclei on the inside. It does not yet produce two complete new daughter cells. So mitosis needs to be followed by another process that's called cytokinesis. Cytokinesis is where you divide everything else. The cytoplasm, you divvy up the organelles, and you split the plasma membrane to separate the two daughter cells apart from each other. And this is a really important concept, right? If you only had mitosis, all you do is you just create those two new daughter nuclei, but without cytokinesis, you can't create two new daughter cells. Now, cell division is not the only thing that a cell does. A cell will spend the majority, really, of its lifetime doing other things. So let's take a look at the entire cell cycle. In the upper right-hand corner here, we have the cell cycle represented kind of like a pie chart. And everything here in this sort of green-blue color, this part of the cell cycle is called interphase. This is where the cell, again, spends the majority of its time. And here, this tiny little sliver of the pie chart in yellow, orange, and this is actually the phase of active division. So this is called the mitotic phase here. Mitotic phase includes both mitosis first, and then that's followed by cytokinesis, right? So in mitosis, you have to create the two daughter nuclei first, and then you follow that up with cytokinesis to form two new daughter cells. From here, each of the daughter cells goes into a new cell cycle. It starts with first interphase and then goes again into the mitotic phase, perhaps. Now, let's talk about interphase for a minute. Interphase is actually divided up into three different sections. So as soon as the new cell is generated, the first part of interphase it goes into is called G1. It was titled G1 because we thought it was kind of a gap in the cell cycle where nothing really happened. We now know that really it's more of a growth phase. The cell grows, it builds up its internal organelle structure, and it begins to function as whatever cell it was destined to be. So for example, if this were a skin cell, it would now behave like a skin cell and accomplish all those jobs that we expect skin cells to perform for us. During G1, the cell makes a decision to either divide or not to divide. If it does decide that it's going to divide, then it's going to enter the second stage of interphase, which is synthesis, right? So the S phase, the S stands for synthesis. This is where the cell says, okay, I'm going to replicate, and so the first thing I need to do is I need to copy all of my DNA. And so essentially a Xerox copy of the entire genome is created during that S phase. Once S phase is complete, then the cell goes into the last third of interphase, which is called G2. Again, this was titled second gap. We thought nothing really important happens here. This is also untrue. The cell goes through another growth phase, and it makes sure that it prepares all the materials necessary for it to go through another round of division. All right, so interphase, you've got G1, S, and G2, and then the mitotic phase includes both mitosis and cytokinesis. All right, now mitosis itself, this division of the nuclear material to make two new nuclei, is divided up into subphases. We're going to go through these briefly on the next few slides. Probably more helpful will be the animations posted on Canvas. Check those out. It gives you sort of a flow to the process of mitosis. But on this slide, for example, you've got actual fluorescence microscope images of a cell going through the different stages of mitosis on the top, and then on the bottom here we've got cartoon diagrams. So the cell will start here in G2 of interphase. While you can't tell just by looking at the DNA that it's in G2, you won't be able to identify whether the DNA is duplicated or not. You can tell that the cell is ready to go into mitosis because it has duplicated its centrosomes. So typically, a non-dividing cell only has one centrosome. But in preparation for division, it has to duplicate its centrosome. Once it's ready, it's got its DNA duplicated, 2 centrosomes, and it has all the material necessary to go into active division. The first stage of mitosis here is prophase. In prophase, a number of important things happen. First of all, the DNA is going to start to condense to show you those sort of more discretely shaped chromosomes. On top of that, the cell starts to build its mitotic spindle. The mitotic spindle includes the centrosomes and these microtubules that extend from them. And this entire mitotic spindle is going to be important for separating the chromosomes during mitosis. Prophase then leads into prometaphase. In prometaphase, we finish a lot of things that we started in prophase. So for example, the DNA finishes condensing. The mitotic spindle is completely constructed, so both centrosomes have now moved to opposite poles of the cell, and they've extended these long microtubules towards the center here. And the microtubules, after the nuclear envelope sort of chunks up and moves out of the way, the microtubules have access to bind to those duplicated chromosomes so that they can start to pull them apart. Now, the microtubules, as they attach to the duplicated chromosomes, they start to sort of yank them from side to side. And this sort of back and forth tug of war motion helps to line up all of the duplicated chromosomes right in the center of the cell here during metaphase. And so this plane, this sort of imaginary plane in the middle of the cell where all the duplicated chromosomes line up, this is called the metaphase plate. And that's essentially the end of metaphase. As soon as all of those chromosomes are nicely and neatly lined up, the cell can go into anaphase. So in anaphase, think A, anaphase, A apart. This is where the cell will actually pull apart the 2 sister chromatids of a duplicated chromosome. Remember that as soon as we pull apart those chromatids, we call them now chromosomes. And so for a very brief period of time here in anaphase, all of a sudden the cell finds itself with double the number of chromosomes. Once the chromosomes are pulled apart and moved to opposite poles of the cell will enter telophase. Now, so telophase is pretty easy to remember because whatever you did in prophase and prometaphase, you do the opposite in telophase. So the chromosomes are going to start decondensing. You're going to start taking apart the mitotic spindle, and you're going to begin to reconstruct the 2 new daughter nuclei on either end of the cell to envelop the newly separated chromosomes. You'll also notice that in this last stage of mitosis, this is where cytokinesis can overlap with the very end of mitosis. And so cytokinesis here, remember the splitting of the cell into 2 daughter cells, you can see this in this animal cell here with sort of this pinched in region. You can see how they're going to separate into 2 individual cells. Again, I encourage you guys to check out the animations that are on Canvas. It gives you a nice flow to this process. Watch them a couple of times and that will help you understand a little bit better the sequence events that takes place in each of the 5 stages of mitosis: prophase, prometaphase, metaphase, anaphase, and then telophase. Telophase, remember, overlaps with the process of cytokinesis to eventually split the parent cell into 2 new daughter cells.